Hi, in this video, we're going to explore the metabolome of bacterial cells. We'll start this process in the phylometabolomic trees tab. We'll notice on this tab, we start with two different types of entry options. One is for broad categories of triplets, as well as the ability to add individual triplets. In our case, we'll go ahead and start with simply the species being bacteria, and we will enter in an organ after that, where we note that the selections that appear after we enter elements in other portions of the triplet are constrained in order to show just valid options. After selecting categories that we want all bacteria cells, we'll go ahead and determine the valid metadata triplets associated with this combination. We will validate this, recognizing that in general we want all of these types of bacterial cells. But we'll also note a drawback of using these ontologies, which is that things that the ontologies group together logically may not be what we're interested in. So we'll go ahead and remove the extracellular fluid as something that we don't want to explore. We will then perform the phylometabolomic analysis, e.g. make our trees. From this, we see the figure that is similar to that in the manuscript, where each element on the x-axis is a type of compound. Each element on the y-axis is a metadata triplet, and the cell value in this matrix is the percent presence. So we can take a look at this diagram and identify different types of metabolites that may or may not be interesting to look at. For example, we see primary metabolism metabolites in nearly every species of bacteria. We see strips of metabolites that are unique to certain types of bacteria, and even types of bacteria that are comparatively distinct compared to other species. For example, this methylamonas gets its carbon from methane metabolism. If we look at this species, we also notice, for example, the compound squalene, which is something that was traditionally harvested from shark liver as its source, but there's great interest in alternative sources. So we can explore this a little bit further. And to do that, we will go to the sunburst tool in this case, and we'll notice that by default, the sunburst tool is searching the compound whose bin identification number is 2, but in our case, we're actually interested in squalene, so we can go here and we can search for that compound. We see that squalene is present certainly in human adrenal glands, very much so. It's present in mice, in different brain and fecal tissues, certainly in the rat feces, certain types of plant leaves, et cetera, et cetera. So maybe it's coming from plants, and it's actually being excreted from animals that eat those plants. Well, if we want an alternative source, we would probably want to use the organ from the plant itself rather than the feces. So one way to simplify this is to use the different types of organs that we're interested in only. So we can use the filtering of this data table to give us a simplified version of the sunburst diagram, where we can look at the different species in which it's found in great abundance. Gossypium actually is cotton, which is obviously very abundant in the United States compared to something like, I believe this is peppermint. This is a type of walnut, and so on and so forth. And so, one way to use this tool is to identify cotton as an alternative source for squalene.